So this micro walk and talk and mock is on the immune response, and particularly going to look at some of those more challenging questions. I won't pick them all out. As always, speed me up, slow me down on YouTube if I talk too fast. Right. It's really important that when you're revising and using your objective sheets, as we asked, that you look at every kind of word actually given. So we're talking about primary non-specific defences against pathogens, blood clotting, wound repair, expulsive reflexes, mucous membranes. So these are things actually that can form part of answers as well. Explain the structure and mode of action of phagocytes, including neutrophils and antigen-presenting cells. Roles of cytokines, opsonins, and phagocytomes and lysosomes. So there's little bits here that not everyone always addresses. They need to be there. And remember, all of these things are listed in that second column on the specification. It says explain the significance of cell signaling, reference interleukins, clonal selection and clonal expansion. And when you're marking this work, I want you to just take note of how many times these key words pop up in the mark scheme. Right, I'm not going to pick all the questions out, but just a little pointer here. We, in terms of our maths skills, we always think we can do the maths, but this is a clear assessment of your GCSE maths, that you know the volume of a cube, a ball, or a sphere even. All right, I'm going to jump down to some of the more challenging questions. This one says, diagram of a stained blood smear observed under light microscope is shown below. If you're not thinking, you might think this is a cell with different parts of it. You might think this is a nucleus or something silly. Fortunately, it's multiple choice. However, this is different blood cells. So looking around, we've got our different ones. I think we've got so we've got some red blood cells somewhere. But I'm going to go with that being our red blood cells because there's no nucleus. I'm going to go with this representing platelets. And then we've got both these two being being, I guess, the type of white blood cell, but we need to consider which one. And both have large nuclei, although distinctly this has got a lobed nuclei. Okay, its nucleus is in several parts. Which of the structures labelled A to D is a neutrophil? So remember that objective? Structure of neutrophils, here it is, and we've already said it, it's the lobed one, so that is B. Okay, so this one says outline the processes that lead to the production of antibodies against an unfamiliar bacterium, and it's 3 marks. This is a clear description. In a way, this is a simpler answer, but I just need to be ultra careful as I'm answering. I don't kind of leap in there with kind of the answers that are too shallow. So what is production of antibodies? So bacterium contains, I use shorthand, so it's a little bit neater because I take a lot of space up and also to save a little bit of time. So bacterium contains antigens on its surface. These are recognised by helper T cells. They're also kind of recognised by B cells directly. So when the production of B cells, and once those B cells, so the B cells will they'll form plasma cells because it's not the B cell instantly. That will start to produce those. And what's that process called? Remember the objectives? That's called clonal expansion. I think that's definitely going to be worth a mark in its own right to say the word clonal expansion. Again, production of antibodies. We could sort of based on go down the road of protein synthesis. I kind of think not for this one potentially. I don't probably need to go that far. When I say recognise, just sort of developing an answer, recognise. Sorry, I should have stinked it around here. The antigen is complementary. Seriously, what's going on? To a receptor. So I've now kind of got 3 bits. I've got the recognition, and I've stated which cells. I've got the process of the B cells from plasma cells, and the antigen is complementary to the receptor. I could mention as well the helper T cells activate the B cells. Seriously. Let's go back. Right. Then it says explain how helper T cells act to speed up these processes. Right, I need to get a little bit more detail now. So what does helper T cells do? I can use they because it's the only word that was given in the. They release. So remember those other words you mentioned? Release interleukins. So those chemicals kind of activate them. They also stimulate them. So when it says these processes, what's it mean? Well, it stimulates B cells, really, isn't it? Inside process. Is that enough? What do they stimulate B cells to do? To multiply. And it's really important because when you look at the mark schemes, and I'm going to jump to mark scheme for this one now. What you need to recognise is that these commas are here. So that's actually really important. Each of these commas is really important. So B lymphocytes have a receptor that carry the antibody on the surface, which means you need to state B cells, antigen receptor, surface, specific complementary to one antigen. You need all of that for the mark. So if I actually go back and review my own answer now, we'll probably find it's not quite detailed. This is 6 pi I. 6 pi I. So we need to say that the B cells, so that's here. Sorry, was it? So I said vaccine with antigens. Okay, well, recognised by helper T cells. That recognition is not enough unless I say there's a receptor. Antigen is complementary to the receptor, so I can say I've got that bit. Is it on the surface? So no, I haven't mentioned that receptor is on the surface of the B cell. So I would not get the mark for that. When you're marking your own work, it's really important you look at that. Because the examiner's going to mark you down, so you might as well now practice writing it in full. Remember, I'm not here to give you the right answers, I'm here to show you how to get the right answers. So as you go through that, watch out for that. Again, on the second part as well, if you look at the mark scheme, it says they release cytokines, interleukins is fine, and the other bit is stimulate B cell proliferation. So stimulate B cells. If you've just said that, that's not worth a mark. You need that to multiply or proliferate. So just watch out how you get both marks and how you include all of those commas. Again, just have a look at this one as another example. We've got a neutrophil, when we need to know about these, pathogenic bacterium, and the role of an opsonium during this process. So if you focus on your objective sheets, this is easy, because you'll be able to say the opsonium helps bind the bacteria to the receptor on the B cell, on the neutrophil even. Sorry. Right, I don't think I know why this is disappearing. So I've got a strange case of disappearing text. Right, other than having specific receptors, describe one way in which the structure of a neutrophil is specialised. Okay, I'm going to start, but I mean, I've got some clues, and this isn't a great clue, but I had a clue earlier in an exam. But think about it, it's going to be the same for all white blood cells. All white blood cells are going to release a large number of different chemicals with antibodies or receptors. So I'm going to say, and we've seen it's got that kind of multi-lobed nucleus. So I'm going to say it's got more, so I'm doing a comparison, rough ER for more protein synthesis. So again, I've developed that answer, not just given 1 point, not just more rough ER, I've kind of given it in a little bit more detail. And last one to have a quick look at, because this is one that examiners said people did badly, compare the primary and secondary immune response by filling the tables below. So relative concentration of antibodies produced in a primary response. Remember, this is getting at this kind of graph when you've got time down the bottom. You've got, maybe accept it up here, you've got time down the bottom, and up the side you've got antibodies. So you get this kind of double hump graph. And what we're looking at is what the difference is between that first hump and then that second hump, what the difference is. So, well, in the primary response, we get a relative concentration rate is low, and then in the secondary response, it's higher. Really important we make comparisons. We don't just kind of say one word, so higher. And the relative duration of the response, or how long it will last, we'll again look at how long each of these are. So short and longer. Okay, get that answer in. Right, just picked out kind of a few of the bits in this one, just for you to have a look at and just get an idea of how to approach some of these questions and some of the things to look out for and stay, especially make the link with your objective sheets. Thank you for listening.